announcements. One is that there is a AWPE course that's available to you. This is trail building and invasive species control. If you need a wellness unit and you want to do something biological with your wellness, here's an opportunity to go out into Wildland Woods or back campus and help Brent Smith get rid of honeysuckle. You get wellness credit for the hard work that it is. Another announcement, there is a conference being organized, Talking Amongst Ourselves, a discussion on health, wealth, and community for students of color. This is on, I don't know if you can see this, it's save the date, March 22nd, 23rd. So I think that's Friday, Saturday, the week after spring break. This student-driven initiative hosted by Early College is a multicultural conference that will provide a safe, welcoming environment for students of color to engage in dialogue about important issues, network, and overall strengthen our communities. Okay, so there's that. And I'm sure that's going to be broadcasted. Okay, so today our goal is the auditory system. Today our goal is the auditory system. We're going to do anatomy of the ear, outer, middle, and inner. We will understand mapping of frequency. So I'll be talking about sound frequency, which is of course pitch. High pitch, high frequency, low pitch, low frequency. And how the, and then get into how the inner ear, the cochlea, is a transducer. It transduces sound into electrical signals that our brain can interpret as sound. And we'll talk about the auditory pathway, the neural pathway, just like we talked about the pain or temperature pathway or the blood pressure pathway. We'll talk about the auditory pathway. And then we'll transition to the vestibular system, which uses a very similar pathway, uses a very similar mechanism to determine the position and the movement of our head in space. So, the best way to start this is with a short video, animation-ish thing. Yeah. Oops. Change this. Okay. So 6 minutes. Overview.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch or frequency produce a slower rate of vibration, and sounds of lower volume or amplitude produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament, and the posterior incudal ligament are of particular importance for the pivotal axis. Two structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth, known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the two fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti, called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
Beethoven's Ninth Symphony, second movement, isn't enough to wake you up at this morning? I don't know what will.
Isn't that a bit of irony there?
What? I'm sorry?
Question about
Yes.
about the last bit there.
The what?
The last bit where it's like the nerve impulses.
Yeah,
And it's just a
well,
different.
we'll get to it. This was the
Okay.
overview. Why don't you save your question until after I've had a chance to teach you?
Sounds good.
Okay. Okay, so, you know, this PowerPoint will be available to you on Moodle. And of course, so will the link. And so the rest of the day will be, the rest of this period will be spent unpacking a lot of that detail. All right, so let's do a little bit of anatomy. The external ear, it's got some fancy names. The pinna, also known as the auricle. This part up here is called the helix. You know what this is called? It's called the earlobe. The tragus, this little nib right here. They've all got names. Not all of them you will be responsible for knowing, but I think you will. I will ask you to know pinna. Anyway, the key concepts list is your reference list for the anatomy you need to know. The auditory canal is carrying sound waves to the eardrum, or the tympanic membrane. You probably know that sound is a wave. It's a pressure wave in the air. So a pressure wave in the air is causing a wave of movement of the tympanic membrane. High frequencies are causing rapid vibrations, and low frequencies are causing low frequency vibrations. Then, as the video articulated in pretty much detail, vibration of the tympanic membrane moves the auditory ossicles. That is an anatomical word to know. 3 auditory ossicles, the malleus, the incus, and the stapes. I will accept the English translations of those Latin words, the hammer, the anvil, and the stirrup. So Latin or English. Malleus, incus, stapes, a movement which are connected to the eardrum via the malleus. Movement of the eardrum, movement of the ossicles, movement of the fluids in the cochlea. We'll unpack this in a little bit. And then transduction of those vibrations by the hair cells of the inner ear. Those hair cells are the cells that do the transduction. The video didn't get into the mechanism of transduction the way I will, so I'll build on that. The video has a mistake. The video says that the hair cells fire action potentials. That's actually wrong. Hair cells are a short receptor cell. They don't fire action potentials. They do release neurotransmitter that triggers action potentials in the secondary afferent nerve of the cochlear branch of the vestibulocochlear nerve that carries those impulses to the brain. Okay? Auditory hair cells, like retinal photoreceptor cells, are short receptor cells. The video goes into some ligaments here that I'm not asking you to know about. It goes into some musculature in here that I'm not asking you to know about. This is the key story that I do want you to know. So here is a blow-up of the middle and the inner ear with the cochlea sort of unfurled, right? The cochlea is that snail shell spiral organ. The cochlea is this snail shell shaped spiral organ, but just to see it better, it's been sort of unwrapped. The stapes vibrates in response to a vibration of the eardrum. Movement of the oval, the stapes is in the oval window. Movement of the stapes causes movement of the paralymph in the scala vestibuli, which is connected to the scala tympani. in, so, and we have the names of ducts. We have, oh, I'm sorry, that is the name of the ducts. The scala vestibuli and the scala tympani that are filled with perilymph, a fluid. Vibration of the stapes in the oval window, vibration of the fluids, allowed because the round window allows, remember fluids are non-compressible, so we need a safety valve to release to allow the vibration of fluid. So the round window allows that. Bottom line, waves of vibration in the fluid, waves of vibration in the basilar membrane that is between the two. That's where the hair cells live. I guess just a blow up of the movement, let's not worry about this. Movement of the stapes, movement of the fluids. Same thing. So as the video mentioned, sound is mapped, the frequency, there's a tonotopic map. Just like there is a retinotopic map in the visual system of information from either the fovea or the periphery in the visual cortex. Remember the fovea is mapped most posterior in the visual cortex, and the periphery is mapped less posterior in the visual cortex. A retinotopic map in the visual cortex. There is a tonotopic map of frequencies in the cochlea. So let's go through that. So here again is a cartoon, oval window, scala vestibuli connected to the scala tympani, and a blow up, an exaggeration of the basilar membrane between them. So scala vestibuli, scala tympani, basilar membrane between them. Because of the physical structure of the basilar membrane, the way it is physically organized, with a narrow base that is stiffer than the floppier, wider apex. So this is the base of the cochlea. The base of the cochlea curls around to go to the apex of the cochlea at the tip of the snail shell. So base, curl, apex. At the base, the basilar membrane is narrow and stiff. At the apex, the basilar membrane is wide and flexible, floppy, less stiff. And because of that, and the rest of the structure of the cochlea, high frequency waves preferably vibrate the basilar membrane at the base, and low frequency waves propagate further into the cochlea and preferentially vibrate the basilar membrane in the apex. So the hair cells in the base and the hair cells in the apex are not very different. But the hair cells in the base are the ones that are vibrated by low frequency sound, and the ones in the apex are vibrated by high frequency sound. Okay? With some preference. And the range, so humans have a pretty good range of hearing compared to other animals. So here's middle A, for those of you who are musicians. You've heard that pitch when you go to a concert, that's what the orchestra tunes to. So let's play some low frequency sounds. I don't even know what note this is. You hear that?
Good.
buzzy because the audio is not great. That's 60 hertz. That's 60 hertz. Turn this down because it's going to hurt. So you can still hear that, right?
Unfortunately.
You can still hear that. Can anyone hear that?
What?
Okay, so this is now, we're demonstrating age-related hearing loss. Because I can't hear anything.
Really?
I cannot hear anything.
No, this is very distinct.
really loud.
Yeah, actually hurts.
Really loud.
So there, you may know that in, I don't know if it's still going on, but where convenience stores are trying to discourage teenagers from loitering outside the store and causing mischief.
Oh no.
They broadcast tones like this at high volume and high frequency, knowing that it'll chase the teenagers away, but their paying patrons will come and go.
I should have really been hearing.
And some of their paying patrons won't even hear it. Okay, so that's the range of, I mean, okay, so you can still hear 7,000 hertz?
Yeah. Yes.
No. I mean, oh, but you can't if you turn it up.
I can hear it. You shut it really well.
I shut the sound off? Oh,
I still hear it.
no, it's still going. I have to press the stop
I
button
think
for
you
it to
did say
stop.
that.
Anyway,
I swear I hear
the nominal range is up to 50 kilohertz, and probably the audio here is inadequately tuned to actually, I'm not sure it's playing 20,000 hertz, right? I mean, it's not a high fidelity system. Okay, so that's the range. That's the range. Okay, yeah, this is the slide we were looking at. Okay, so low frequency sounds are mapped to the bass, and high frequency sounds, high frequency sounds are mapped to the bass, low frequency sounds get to and are mapped to the apex. Intermediate sounds are mapped intermediate. Intermediate frequencies are mapped intermediate. So the tone is mapped. The amplitude determines how big the vibrations are. Was this the one we were looking at? This is the one we were looking at. This is the same in, yeah, okay. So here is a cross-section of the cochlea, scala vestibuli, scala tympani. This is the basilar membrane that holds the organ of Corti, that holds the hair cells. So let's go through the anatomy again, a little bit. Oops. Scala vestibuli, scala tympani are filled with perilymph. This cochlear duct is filled with endolymph. There is a membrane here that I'm not asking you to know the name of. In fact, I'm forgetting it myself. This is the basilar membrane that I'm not asking you to forget the name of. I'm asking you to remember the name of. The basilar membrane holds the organ of Corti. That is a name to know. The organ of Corti is structured like this, with the basilar membrane between the tectorial membrane. This is all part of the organ of Corti. The basilar membrane, the tectorial membrane, and the hair cells between them. Deflection movement of the basilar membrane causes bending of the stereocilia, which the video called hairs. They are called hair cells, but this is not hair. This is not hair. These are stereocilia. Let's look at this in blow up. Basilar membrane, hair cells in a supporting matrix. Hair cells here and here. Inner and outer is not a thing that I need you to know. They all have stereocilia that are in contact with the tectorial membrane. And so when there is a deflection of the basilar membrane due to a vibration propagating into the cochlear duct, the scala, vestibuli, and the scala tympani, and movement of the fluids, movement of the basilar membrane. Let's just take one deflection. Deflection up causes a shift in this position and a bending of the stereocilia. The bending of the stereocilia is where auditory transduction is going to take place. That's going to be key for converting a pressure wave sound into an electrical signal. The hair cells and the bending of the stereocilia are what do that. Yeah.
So like once they bend, is that when
Yeah, you'll see that in a minute. I'll take that apart. So here is a cartoon and an electron micrographic photograph of the hair cell, a hair cell, right? So it's a cell, it's got a nucleus, it's got all the things that you learned about in cell physiology, being ignored here. It's in a supporting matrix. It's a short receptor cell that does not have an axon, but it does release neurotransmitter. It's being cartooned here with this daisy thing, right? Neurotransmitters that are loaded with neurotransmitter, docked at this membrane, ready to be released. What do you suppose causes the release of this neurotransmitter? It's the same old, same old. Calcium influx, calcium binds to synaptotagmin, snare conformational changes, fusion, release of neurotransmitter. It's an excitatory neurotransmitter, simple excitation of the afferent nerve. Now we create an action potential. So the movie has something wrong. It says the hair cells make action potentials. The hair cells don't, but they trigger action potentials in the afferent nerve.
Why is there that inferred axon there?
I'm sorry?
That axon there on the right.
So why do we have an efferent axon? Because we can tune, so this is the afferent axon going from the hair cell to the brain, carrying auditory information. We have efferent axons that bring information from the brain and from the body actually to tune the hair cell. So, for example, if we're in an environment that is very loud, we can try to stiffen the hairs and prevent them from moving quite as far in order to protect our ears from the damage caused by loud sound. We have some protection, and that's part of what's going on here. But this is not the key story for auditory transduction. This is a modulatory thing that I'm not asking you to know. This story about the hair cells, the cells, neurotransmitter release, and the afferent neuron is what I would like you to know. So here are images of the hair cell bundle, sort of from the side, from the front. So this cartoon is pretty accurate in the sense that there's a stair-stepping of length of the stereocilia of the hair cell. So this is looking at it face on, short, longer, 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 longer. stereocilia, a well-organized pattern of short ones next to longer ones, next to longer ones, going up to the longest one.
Is there a physiological mechanism that keeps the hair, like the stereocilia together, like as a bundle?
Is it, that keeps this organized like this?
Yes.
You know, it's pretty, there is, the endolymph, the stiffness of the hair cells, and the stereocilia, these are fragile structures. And this is partly why we have age-related hearing loss, is that they can be damaged. They can be damaged by excess vibration, so playing loud music can damage them. An example of that. Certain drugs can cause toxicity. Hair cells are susceptible to drug-induced cell death. So there are certain drugs that kill hair cells and cause hearing loss. Yeah.
I've heard people say that they don't like the dangers of headphones like this as compared to like having just loud sounds and big speakers around you. Headphones. I said headphones, right? Not
Yeah.
headphones. Cool. Is it more dangerous to have like a source right next to your ear than to have a source that's farther away but really loud?
I think it's easier to get too loud with headphones than it is to get too loud with speakers, because with speakers, we are aware that we're disturbing our neighbors, and with headphones, we're aware that we're not.
So, what is the predictions for like the relative hearing loss or attenuation as compared to previous generations? It's supposed to be a lot higher.
I don't know. I can only speak for me. I don't know the answer to that. So here's another electron micrograph of a hair cell. So here's the top of the hair cell. Here's the hair cell bundle, stereocilia forming this stair-step pattern. There's a, you may have noticed here in this cartoon, there is a the longest one, which isn't actually the longest one. The structure that the stereocilia are sort of leaning up against is a special one called the kinocilium. So here you can see the kinocilium standing behind the stair step of stereocilia. It's got to be one of the coolest structures in the world. The cells around the hair cell are the supporting cells that hold it in place and support it so that these stereocilia are appropriately embedded in the tectorial membrane, which has been removed in this preparation.
What do you say the kinocilium?
Kinocilium is the name of this one in the back, the tallest one behind the stereocilia. Kinocilium.
Does that play a role in?
Yeah, it's a supporting role. So here's another image of, organization of stereocilia on an array of hair cells. Here's a hair cell, here's a hair cell, here's a hair cell, here's each hair cell bundle, the bundle of stereocilia for each hair cell in an array. So now, let's talk about how we transduce, our hair cells, transduce vibration into electrical information, electrochemical information, because we're immediately going to get to the release of neurotransmitter. When the hair cell bundle, this bundle of stereocilia, is bent away from the kinocilium, which is not shown in this image. Remember, the kinocilium is behind the longest one. So when we bend this structure away from the kinocilium, a structure that connects the shorter stereocilium with the longer stereocilium, called the tip link, because it's linking the tip of the short hair cell the tip of the short stereocilium with the wall or side of the longer stereocilium. That's the tip link. It's a key structure. When we bend it this way, the tip link gets less tension. So here is, let's just short stereocilium, long stereocilium. I'm going to imagine the tip link between the tip of this stereocilium and the side of this stereocilium. Bend it this way. Did you see how the tip is getting closer to the side, and so the tip link tension is decreasing? If I bend it this way, see how the tip is getting further away from the wall and tension is increasing. Slack, tense. Slack, tense. Slack, closing a stretch-activated cation channel that is located on the tip of the shorter stereocilium. They are activated by stretch, so they are activated when the tip link stretches them open. Closed, open, stretch-activated cation channel. It's got a name that is rather opaque, so let's just call it the stretch-activated cation channel at the tip of the stereocilium. I don't want you to learn. The name isn't helpful, so I'm not going to make you learn it. Closing a cation channel allows the cell to polarize or hyperpolarize. Opening a cation channel allows the cells to depolarize, just like you've already learned. So we're using some of the key principles that you've learned. Hyperpolarization allows calcium channels to close. Depolarization allows calcium channels to open. One we're off and running. Closed calcium channels, less calcium influx, less calcium, less neurotransmitter release. More calcium channels, more calcium influx, more calcium binding to synaptotagmin, more snare complex conformational changes, more neurotransmitter release. The neurotransmitter is excitatory. The neurotransmitter actually is still being worked out, but glutamate is probably one of them. Less neurotransmitter release, less depolarization and action potential frequency in the afferent nerve, more neurotransmitter release, more depolarization, and more action potentials in the afferent nerve. And that's where we are, in the afferent auditory nerve. The auditory branch of the vestibulocochlear nerve, which is one of your cranial nerves, is what carries this auditory information to the brain, which we'll get to next. Shai?
So you said the stretch of the tip link is what causes this series.
Stretch of the tip link is what causes this series of events.
Yeah, so is it because the tip link is only linked to the tip of the shorter one, if you think about it in a chain, is it that the stretch always activates the hair cell that is before the tip link? Or
No, I mean,
left of
remember,
it?
this is one hair cell.
Yeah.
So this hair cell, let's go back an image. So this hair cell, when this bundle, the whole bundle, remember, is bent this way or bent this way. When the whole bundle is bent, let's activate it. We're gonna stretch the bundle, we're gonna bend the bundle this way. Okay. Channels, mechanically or stretch activated channels here are opened by the tip links here. Stretch activated channels here are opened by the tip links here. It's all running in parallel.
So can we say that the longest hair cells do not have these?
The longest hair cells probably express the stretch activated channels, but there's no tip link to open them. Yeah, does that help?
That is what it is.
Yeah. And here's an electron micrograph of the structure. This electron dense material is what contains the stretch activated channels, and this attachment here to the wall is a physical attachment that obviously makes a difference. Because if it's an interesting story of being able to use cytoskeletal motors like kinesin and dynein to move this location up and down on this wall of the longer of the two stereocilia in order to, adjust and perfect the tension on the tip link. So this is an adjustable parameter. This is not something you need to know, but this attachment here is adjustable. This attachment here is to the mechanically gated, the stretch activated channels, and this electron dense material includes the channels that cause, remember, if a cation channel opens here, depolarization here, which spreads throughout the hair cell. Let's take a break now, and we'll get to the pathway of auditory information. I'm going to make a correction. I think when I was pointing to this image, I got the scala wrong. This is the scala tympani, and this is the scala vestibuli. Here it is in cross-section labeled correctly. Scala vestibuli, scala tympani. This is the cochlear duct. This is the organ of Corti. This is the tectorial membrane. Here are the hair cells with the stereocilia in contact with the tectorial membrane, bending of flexibility of this structure, bending of the stereocilia, bent towards the kinocilium. The tip links relax, this tension decreases, less depolarization of the hair cell, less activation of action potentials in the vestibular branch, the cochlear branch of the vestibulocochlear nerve, fewer lower frequency action potentials going to the brain. The frequency of action potentials does not align with the frequency of the sound. The frequency of the action potentials is carried in specific axons that are connected to specific hair cells that are activated preferentially by specific frequencies because of the tonotopic map of frequencies on the basilar membrane. So it's not that the frequency of action potentials is connected to the frequency of the sound. The frequency of sound activates specific action potentials, activates specific hair cells that then increase or decrease the action potentials of their connected nerve cell. And then which nerve cells are firing is what carries tone information to the brain. Which nerve cells are firing high frequency action potentials and which nerve cells are not is what carries tone information to the brain. Alex?
How do we know about, how do we know about like loudness and softness?
Loudness and softness, amplitude has information, loud, soft, but that amplitude is the amount of neurotransmitter released by the hair cell. And the frequency, so the frequency of action potentials and the pattern of action potentials in a neuron actually carries more amplitude information. And which neuron is firing action potentials or not is what carries frequency information from the organ of Corti to the brain. Does that make sense? Is that clear? Go forward. So again, just to state it, so we've transduced sound into frequency of action potentials in specific neurons. Which neurons are highly activated is what carries pitch information. How activated they are carries amplitude information. The auditory or the cochlear branch of the vestibulocochlear nerve, cranial nerve something, 8, I don't know. Is it 8? The vestibulocochlear nerve is cranial nerve 8. So the afferent neurons from cranial nerve 8 carry the auditory information to the medulla oblongata. So where is the first synapse in the auditory pathway? It's a trick question, because I just gave you a name, but that's not the correct answer. The first synapse is in the organ of Corti, between the hair cell and the afferent neuron.
But
That's the synapse, that's the first synapse.
This
The
is-
second synapse is in the medulla. Yeah.
does the action potential even exist in the hair cell? It doesn't.
It's not an action potential, but it is, that is a synapse.
But the action potential only starts after that first sentence.
That's right.
Okay.
The depolarization of the afferent neurite triggers action potentials in the afferent nerve, which carries information a relatively long distance compared to the distance between one end of the hair cell and the other end of the hair cell to the brain, to the medulla oblongata.
So you're saying some of this information crosses over, but some of it doesn't?
Yep, some of it crosses over and some of it doesn't, so I'm not gonna ask you to know this. I do want you to know that the second synapse in the auditory pathway is in the medulla oblongata. There are some other synapses in the inferior colliculus. Remember, we talked about the superior colliculus in the visual pathway for coordination of eyeball movement. Now we have the inferior colliculus in the midbrain processing auditory information, and it's both contralateral and ipsilateral. It's gonna be processed in the thalamus, so remember the lateral geniculate nucleus of the thalamus is important for processing visual information. The medial geniculate nucleus of the thalamus is important for processing auditory information. Synapses all along the way. So, synapse here, synapses in the medulla, synapses in the inferior colliculus, synapses in the medial geniculate, and then axons from the medial geniculate nucleus project to the auditory cortex. The auditory cortex is in the primary auditory cortex is in the superior temporal gyrus. There is a map of tone in the primary auditory cortex, in the superior temporal gyrus. High frequency is mapped posterior, and low frequency is mapped anterior. Information from one ear relative to the other ear is mapped mediolateral, and I'm not going to get into the details. But I do want you to know that there is this tonotopic map. So just as there is a tonotopic map of frequency in the cochlea, what was it again? High frequency is in the base, low frequency in the apex. There is a much higher processed map of tone in the auditory cortex, with high frequency mapped posterior and low frequency mapped anterior. Posterior, anterior. Okay, so we're gonna do a small transition to localization of sound in three-dimensional space. So we've been focusing mostly on transduction of sound, process or coding of pitch information, and coding of amplitude information, which you've now learned about. But you also know that you can process sound in three-dimensional space. So, and I mean, I'm sure you're aware of this, but just to demo it, I need a volunteer. Any volunteers? All you have to do is stand here.
You can volunteer as yeah, you have the best feeling hearing here anyway, so.
You gotta stand here with your back towards us. You don't have to keep your eyes closed, but you do have to not be looking at us. And what I want you, let me give you the instructions before I do the test. When I make a sound, I want you to point, you know, it's going to be hard because you're pointing backwards, but point to the general location of the sound. Okay?
Assuming everyone else has to be quiet.
Pretty good, right? I mean, it was hard for, you're done. Thank you very much. He was pretty good at being able to identify behind him, which is actually what we're involved better in the front than we are in the back, but he was able to get a pretty good sense of whether it was over on this side or right behind him or over on that side. It wasn't perfect, but it was pretty good. Let's talk about how we do that. There are two mechanisms. One is, so we're talking about localization of sound in the horizontal plane. We're not going up and down, we're going horizontal. How did Tariq know that when I clicked my fingers over here, it was over here, even though he wasn't seeing me? The first mechanism is an incredibly sensitive mechanism that allows us, because we have two ears on either side of our head, which is 8 inches or something. I don't even know how many inches are between my two ears. More than 6, it's not a foot, so something like that, right? The difference in the location of my ears tells me information about where the sound is coming from, because we can detect the time difference between a sound hitting one ear and hitting another ear. Sound has a speed, right? It travels at 600 miles an hour or something like that. So it takes a tiny fraction of a second longer for the sound to reach one ear than to reach the other ear if the sound is off the center. If the sound is right in the center, it's reaching our both ears at exactly the same time. So Jacob's both ears are hearing that sound at exactly the same time. Now his left ear is getting that sound a tiny fraction of a microsecond, or a fraction of a millisecond. It's actually measured in microseconds sooner than the sound is reaching his right ear. Does that make sense? And we have an exquisite mechanism for detecting that difference in sound. So let's see if we can walk through this. I've never had the time to go through this in great detail, and let's try it. So here is a sound reaching the left cochlea first. So this is a sound that is closer to my left ear. but it reaches my right ear a little later. By little, we're talking about less than a millisecond later. And you can do the calculation. If your ears are 6 inches apart and sound is traveling at 600 and something miles per hour, how many fractions of a millisecond are we talking about? You can do that calculation. So action potentials are initiated along the auditory nerve of the vestibulocochlear nerve, auditory branch of the vestibulocochlear nerve, and then make synapses in the, this is a cartoon of the nucleus in the medulla that is receiving the second, this is the second synapse. First synapse was here, second synapse was here. This nerve makes a series of synapses in different cells in the medulla. This cell is doing the same thing. Same type of synaptic connection. This action potential is reaching all of these synapses in an order. It's reaching this one first, and this one second, and this one third, and this one fourth, and this one fifth, because of the length of the axon necessary to carry it, right? I mean, you know that nerve action potential propagation is something like 100 meters per second. And so if this is a millimeter apart, this is getting the, I'm not going to do the math, but this is getting this release of neurotransmitter here a fraction of a millisecond before this one. And this one is receiving this one is doing the opposite. I mean, it's doing the same thing. It's doing exactly the same thing. It's getting this one a little bit sooner than this one. We process this location by which of these cells receives the two information simultaneously and maximally activates this third order neuron. So if the cell is right in front of us, this cell would be maximally activated. And if the sound is coming from the left, the rightmost, the sound is coming from the left, I don't remember. A different cell will be receiving the maximal activation. And if the sound is receiving from the right, the rightmost cell in the medial superior olivary nucleus, not a name you need to know, of the medulla, a name you do need to know, is maximally activated. So which of these cells in this nucleus of the medulla is maximally activated is what we learn to code for, oh, the sound is from over there. You know, as babies, we start to learn, oh, when mommy's calling from over there, I'm going to turn over there. Yes, Aiden?
So does this have to, the timing has to work out such as you have the same neuron being activated pretty much at the same time by multiple action potentials?
That's right.
So if
Exactly. So this simultaneity of input from two different neurons is what's maximally activating cell E.
And if it wasn't exactly simultaneous, then would you
Right,
not
so-
have that?
Well, that's right. So, I mean, this is just 5 cells. There's probably a few thousand cells in here that are allowing us to tune the which of the cells are maximally activated, that is to say, which are receiving the information simultaneous from both ears. If it's the ones in the middle are receiving simultaneous information from both ears at exactly the same time. That the one on the left is receiving simultaneous information from both ears at exactly the same time. And what we're doing is we're coding, if it's in the middle, it's from right in front of us. If it's from the side, it's from the side. I mean, if the cell that is maximally activated is from the side, we know, we've learned, that's what tells us the sound is from over there. Or that's what tells us the sound is over there. Or that's what tells us the sound is straight in front of us. Yeah.
So how do we know if it's behind us?
Well, same mechanism. So Tariq was illustrating that we can do this So a sound from directly in front and a sound from directly in back can sometimes sound exactly the same. So sometimes we get confused. I don't know, I mean, we didn't do it with Tariq because he knew, his brain was telling him, well, it's gonna be from behind me. There's no way the sound was gonna come from that direction when he's facing this way. So, but if you're standing out in the middle of a crowded room and somebody calls your name from directly in front or in behind you, sometimes it's, you're looking for that person. You know it's either here or here, but you're not sure which. The other way, so one mechanism, let me just summarize. One mechanism is the timing of information from the two different ears, receiving or getting to the medullary neurons at slightly different times. Which of the neurons receive the information simultaneous from the two ears? So the two ears are receiving information at asynchronous times, but because of the difference in the length, some cells and not others are receiving the information at the same time. And that's what's coded, that codes orientation of sound in three-dimensional space. Well, in the horizontal plane. It's not how we do this, as you can imagine. The other way we process the location of sound is by a very sensitive manipulation of sound by our outer ear, by the pinna and the tragus and the auditory canal. We have learned, so, and you don't need to know this, but the point is the sound sounds a little different whether it is coming from straight in front of us, at an angle, or on one side. So we're not talking about the timing of the sound, we're talking about the sound of the sound. This is a spectrum. Low frequency, high frequency, relative amplitude. Sound sounds a little different if it's straight in front of us than compared to if it's way on the side. And we've learned as human beings to interpret these spectra of sound, and we can convert that into localization. So you don't have to know the details of this, but you do have to know the two mechanisms for localization of sound in the horizontal plane. This one is actually what we use for the localization of sound in the vertical plane as well. Sounds coming from above us, directly above us, which are reaching our both ears at our same time, give us no information about whether it's, the timing gives us no information about here versus here versus here, but we have learned how to process the sound of the sound to know whether it's coming from above us or straight in front of us. I haven't done this. Let me have another, oh no. Well, yeah, let me have another volunteer. Let's try this.
Get up there. Do. Come on. Your pressure. Go for it.
So this time I do need you to close your eyes
Like
and you face us. I want you to point to where the sound is coming. So he can do it in three-dimensional space. Very good. Thank you very much. Okay, now we're going to switch gears again and talk about the vestibular system, which is not really part of the auditory system, but uses the same organ, the inner ear, and uses the same mechanism, hair cells. So here's the cochlea that we've just been focusing on. Here's the cochlear branch or auditory branch of the vestibulo-cochlear nerve, cranial nerve 8. Now we're gonna be talking about information carried in the vestibular branch of the vestibulocochlear nerve from the other part of the bony labyrinth, other parts of the bony labyrinth. So the vestibular system has, well, several major components. I'm not going to count them out. Let's talk anatomically, let's talk about them. So there is a organ, a small suborgan called the saccule, which is a bulging of this bony labyrinth closest to the cochlea. Then further away is the utricle, and then further away still are these very obvious semicircular canals. And this is a cartoon in two-dimensional space, of course, but the semicircular canals are in 3 dimensions. One, so a circle, so imagine this one is sort of oriented out like this. This one is oriented back behind the screen a little bit, and this one is, it's circular, but it looks oval because it's close to horizontal. So let's just simplify that. Dimension 1, dimension 2, dimension 3. 3 different dimensions in the semicircular canals. In the semicircular canals, there is a structure, a bulging called the ampulla, and inside the ampulla is a structure called the cupulate. This is where hair cells are in the semicircular canals. And we have a similar structure inside the saccule and the utricle. So these structures allow us to determine the movement of our head. It's not transducing sound, it's transducing movement of our head in 3-dimensional space. So let's just see how this is done. So this is a blow-up of a ampulla and a cupula of a semicircular canal. There is fluid inside the semicircular canal. The cupula has hair cells very similar to the hair cells in the organ of Corti. They bend in response to the movement of the fluid. But the movement of the fluid is not due to the vibration of sound, it's due to the movement of our head. So when I turn my head to the left, the fluid, which has inertia, is moving to the right relative to the movement of my head, right? The fluid has inertia. And so if I move my head to the left, the fluid stayed still, which meant that relative to this cupula, it moved to the right. Let's not worry about the directions. It bends the stereocilia. And that bending of stereocilia carries information, bending of stereocilia, depolarization, activation of calcium channels, calcium influx, synaptotagmin, release of neurotransmitter, activation of afferents from the cupula to, in the vestibular branch of the cochlear nerve, and it's preferentially, in this case, going to the cerebellum. So if I move my head, so this is giving my body information about the movement of my head in space. If I move my head to the left, my body is making adjustments for the fact that I've moved my head to the left. When I move my head to the right, same thing. I'm rotating my head down, I'm rotating my head up, I'm rotating my head to the side, right? I'm moving my head in 3-dimensional space. I'm rotating my head. And that's activating hair cells in the cupula of semicircular canals. And if the semicircular canal is aligned with the movement of the fluid, it's causing a big deflection. of of the, so in this case, it's the semicircular canal that is oriented this way. Or this, I've got three-dimensional movement, and that's activating the semicircular canals preferentially, giving my cerebellum information about the movement of my head. Yeah.
So is it cupula, like, is it a structure, or would you like classify it as kind of like a bundle of hair cells, like at the ear cell?
The cupula is the structure that holds the bundle of hair cells, sort of like the way, well, there's probably a name for the structure in the organ of Corti that holds the auditory hair cells. So the ampulla is the equivalent of the organ of Corti, and there is a name here, the cupula, which is what holds the hair cell bundle.
So the stereocilia is inside the cupula? It's not
The
same?
stereocilia are really inside the cupula
Okay.
in contact with the fluid. And the
Okay.
hair cells, the cells themselves, are embedded in this structure called the cupula. All of this is in the ampulla.
Okay, thank you.
Yeah.
So you said that those discs, the semicircular discs or whatever, sort of, what?
Semicircular canals.
Canals, I'm sorry. They sort of correlate with the XYZ plane?
Kind of, but not perfectly so.
So like, is it, so I take it in the data is kind of sort of compared between the discs?
Sure. So if I move my head in the horizontal plane, I'm activating the one that is closest to horizontal, but I'm also activating some of the others, and my brain has learned to interpret that as movement in the horizontal plane.
If someone had perfectly X, Y, and Z oriented semicircular ducts, would that be beneficial or would that limit their hearing?
It wouldn't limit their hearing, but apparently is not beneficial enough for us to evolve it. Can't be much better. So now let's finish with, we're not gonna do this, we're not gonna have time for our game, but let me remind you, well, so the semicircular canals give us information about the rotation of the head. The utricle and the saccule give us information about acceleration in two-dimensional space. So when I take a step forward, I've just accelerated my head in this direction. When I take a step backwards, I've accelerated my head in this direction. When I take a step sideways, I've accelerated my head this way. So the utricle and the saccule report acceleration, movement of my head in straight lines. And the semicircular canals tell me information about the rotation of my head in three-dimensional space. Kendra?
Is the cupula a short receptor cell, or?
All hair cells, no, cupula is not a cell, cupula is a structure. The hair cells here are short receptor cells, just like the hair cells in the organ of Corti. Here are this is supposed to be a cartoon of the afferent neurons, so the first synapse in the cupula, the first synapse in the vestibular system is in the cupula. Our cerebellum is what's important. I'm not going to talk about the pathway because it's really complicated, but this information, rather than going to the auditory cortex, goes to the cerebellum first, and then is processed by the cortex.
So this is right next to the cochlea, right?
It is right next to the cochlea, so that's why it is called the vestibulocochlear system, and the vestibulocochlear nerve is what happens after these nerves merge together into one.
Now, my dad, when he lifted something really heavy and tore some part of his inner ear, and his balance was really off
Yeah.
for a while.
Right,
And he said he also heard the sound of like a hissing steam pipe. So I'm wondering how.
okay. So the hissing steam pipe is called tinnitus, and that's probably damage to the hair cells. The balance could be damage to the hair cells in the vestibular system, or it could be damage to the vestibular nerve. So let's see, I just want to finish up. There's a series of this is the Bing game that we're not going to have time to play, but you can play this with, with your friends. I just want to make it clear that some of the names on the Bing game are names that I do want you to know, like cochlea. And some of the names on the big game are names that I don't want you to, that I don't ask you to know, like tensor tympani muscle. Use your key concepts list to know which is which, but there are some of these. It's fun, gives you that pleasant ding sound, think about the frequency of that sound. But like oracle and auditory canal and incus, those are names that are on your key list, and so you can play the game to help reinforce your knowledge of the auditory system. Okay, I'll see some of you in lab this afternoon.